Welcome back, everyone. So we are going to dive into our fifth unit of the term. So this unit dives into how cells obtain energy. So we're going to take what we learned last week with energy and metabolism, and we're going to focus in on basically cellular respiration this week. Now this can be a complicated topic, so we're going to work our way through it very carefully, break it down so that we understand it completely. So this is chapter 5 in your textbook for the OpenStax. If you are using the Campbell Biology textbook, it would be chapter 7. So we have it broken down into four sections. So we're going to basically be talking about cellular respiration, which can be broken down into two segments here. The first segment we're going to talk about in this video, glycolysis, and then the second section is going to cover the other two parts of cellular respiration, which occur in the mitochondria, the citric acid cycle and oxidative phosphorylation. We're also going to learn about other ways that cells can obtain energy aside from cellular respiration. So we're going to learn about fermentation, which is another method that cells can basically bring together ATP. And then we're going to look at connections to other metabolic pathways. So throughout this chapter, again, we're learning about last week we learned about how living organisms, they use energy, right? So now we're going to learn about how they obtain energy. So how they take their fuel, that food, that glucose molecule, and how they can break it down and actually extract energy from it and utilize that energy. Okay, so we'll learn about that this week. So by the end of this video, we're going to again learn about glycolysis. And so by the end of the video, you should be able to explain how ATP is used by the cell as an energy source. You should also be able to describe the overall result in terms of molecules produced by the breakdown of glucose by glycolysis. So when we think of ATP, you always want to think of it as energy currency for cells. Okay, so this is, what the cells use as an energy source. Okay, so it allows cells to store energy briefly and transport it within itself. It supports those endergonic chemical reactions that we learned about last week. So remember endergonic. Those are those reactions that require energy. So we'll learn about the structure of ATP and how ATP, when we break off that phosphate group, how energy can be extracted from that and how we can harness that through the process of glycolysis. So let's start out by talking about what ATP is. So ATP stands for adenosine triphosphate. So adenosine triphosphate, ATP. So that is used to power the majority of energy requiring cellular reactions. So if we take a look at this reaction right here, ATP plus water, right away, what type of reaction is it? Is it a dehydration synthesis reaction or a hydrolysis reaction? When we're adding water to break apart ATP. Well, this would be an example of a hydrolysis reaction. Remember, hydro means water, lysis means breaking. So we're using water to break apart the bonds that bring ATP together. So again, we're kind of reviewing, hey, you have a midterm coming up, so we're reviewing some of those terms that we learned earlier in the term. So this is an example of a hydrolysis reaction. So water is breaking apart ATP. Okay, and in doing so, is this an example of an anabolic or a catabolic reaction? So this would be an example of a catabolic reaction because we're breaking apart a large molecule and releasing free energy. Okay, so remember catabolic cats, they break things and release energy in the process. So this would be an example of a catabolic reaction. And finally, is this an example of an endergonic or exergonic reaction? So this is an example of an exergonic reaction because we're ending up with releasing energy in the process. So we start out with more energy than the products have left behind. Okay, so we've gone through all these terms already. Okay, so we have ATP, adenosine triphosphate. So we're adding water to this reaction. So we're breaking apart ATP. We're removing one of the phosphate groups. And in doing so, we've dropped down to ADP, adenosine diphosphate. So we've released this phosphate group. And by releasing the phosphate group, we've released energy. Okay, so that's why it's an exergonic reaction. We've released energy. Okay. Now this, the reverse of this reaction is an endergonic reaction. We have ADP, so adenosine diphosphate. If we add a phosphate group, okay, to this reaction, and some free energy, we have to add that. So we're adding energy to bring together the phosphate group with the ADP. We're going to end up with ATP. And in the process, we're removing water from that reaction. So that's dehydration synthesis. Okay. So this is basically you can see that we're going to actually use this as an energy coupling. We learned that term last week with our video, with the crash course video, where we have one reaction kind of drives the other. Okay. So we're going to start out with an Amoeba Sisters video that will talk about ATP and how we can harness energy from it.
ATP. We love it. You'll find ATP in so much of our science art. Whether it's a GIF of a mitochondrion or cell transport, or a comic about cellular respiration or fermentation, you'll see ATP mentioned. So why the big deal? Why is it all over the place? Many times students will get it in their mind that it is an energy currency of some kind. When I first started studying biology, I noticed that in textbooks, it's often represented like this starburst thing or thunderbolt. And, I guess in my mind I imagined it was like some big blast of energy to help the cell do things. And by do things, I mean that we need ATP to do many cellular processes. Examples include active transport, such as when a cell is trying to move something against its concentration gradient, or its role in muscle contraction with the actin and myosin cross bridge. We need another video for that. ATP is critical for many types of cell signaling. You need your cells to be able to communicate. Those are all just some examples. But what is ATP? How do we get it? And how does it work? Those are the basics of what we're going to focus on in this short video. So what is ATP? If you remember the four major biomolecules, ATP would fit in with nucleic acids. Yes, like DNA and RNA. ATP is a nucleotide derivative, so it has those three parts you'd see in DNA or RNA nucleotides: phosphate, sugar, base, but it actually has three phosphates. ATP is short for its full name, adenosine triphosphate. This fancy name is helpful as it tells you that it contains the nitrogenous base known as adenine and three phosphates, hence the tri in adenosine triphosphate. Its sugar is ribose. How do you get ATP? All cells need ATP, and so they need processes that can be used to generate it. But the process can differ. It might involve oxygen, such as aerobic cellular respiration. It might not involve oxygen, such as anaerobic respiration or fermentation. During cellular respiration, plants break down the glucose they made from photosynthesis to make ATP. During cellular respiration, animals break down the glucose they consumed to make ATP. And it's not just plants and animals. Bacteria, fungi, protists, and archaea, they all need to make ATP. We have a video on cellular respiration and another one on fermentation that can be helpful to understand the process. But one thing that we do want to mention about making ATP is it's important to understand it is part of a cycle. With the ATP cycle, you have ATP, which can be hydrolyzed, releasing energy and losing one of its phosphates in this process. A process like cellular respiration can provide the energy needed to add a phosphate to ADP in order to regenerate ATP again, which is important as ATP can be used quickly. This brings us to how ATP is able to work. So how does ATP work? It's not just about ATP being hydrolyzed and releasing energy, it's more than that. Okay, honestly, it's more than our short video can go into, which is why we provide some further reading links, but let's look at some basics. So when ATP is hydrolyzed, meaning here it involves the addition of water, it's not really that the bond between the second and third phosphate itself is a super strong bond. It's actually more that the bond between the second and third phosphate contributes to this ATP being unstable. These phosphates with their negative charges don't like being arranged like this. The change from ATP losing its third phosphate to become the more stable ADP is an exergonic reaction and releases free energy. A popular example for understanding ATP is to use the spring illustration, like a wire spring. Consider how you might compress the spring. ATP would be modeled by that compressed spring, and then you would let it go until it just goes into this relaxed state, which would be represented by ADP. When ATP is hydrolyzed, if the energy was just released, it will likely not be useful for a cell if it's not actually coupled to something that needs it. Thankfully, the energy release can be coupled to endergonic processes that the cell needs to do. This can occur when the phosphate from the ATP is transferred to a molecule that is going to be acted upon. For example, this cell transport protein here is supposed to move some kind of molecule against its concentration gradient. Recall that if it was passive transport, these molecules would be moving from high to low concentration. But in active transport, thanks to ATP, this protein can move them against the gradient. When the phosphate is transferred to this protein, we say the protein has been phosphorylated. Sounds powerful. We can say in our example that this protein is more reactive and less stable in this form, this phosphorylated intermediate state. When it reverts into its original, more stable shape, it can assist in moving them the other direction. So, from marveling at the beating of a single cilia hair, or chromosomes being separated in cell division, or binding the correct amino acid on the tRNA, I could go on. We hope that little ATP symbol will mean something every time you see it. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so again, that was a good video to kind of introduce us into how ATP is used by the cell, and again, how that ATP undergoes that exergonic reaction so that it will be involved in that energy coupling with an endergonic reaction. So the phosphate group will break off and attach to another molecule in order to provide the energy for that endergonic reaction. So if we take a look at that ATP molecule, remember ATP stands for adenosine triphosphate. And from the video, it referred to ATP as a nucleic acid. And the reason why it's a nucleic acid is because it contains a nitrogenous base. So here's adenine. So recall from our macromolecules talk, we have our nucleotide that contains a nitrogenous base, a sugar group, and phosphate group. So in this case, we have adenine as the nitrogenous base, and then you can see that we have ribose is the sugar. And ribose, hey, that's the same sugar that RNA has, right? So that's that 5-ringed or 5-carbon ringed sugar. And so same as RNA. And then if this was AMA, adenosine monophosphate, it would have one phosphate group. Okay, so that would be a nucleotide. And then if we add a second phosphate group, it's called adenosine diphosphate. If we have a third phosphate group, then it's adenosine triphosphate. So think, mono means 1, di means 2, and tri means 3. Okay, so it's a little bit of Latin there, so it's just how we, refer to things. Okay, so adenosine triphosphate means there's 3 phosphate groups, but we'll be dealing with adenosine diphosphate as well. So just think of 2 phosphate groups with one have basically fallen off through that reaction. Okay. All right, so when we do have that, so the goal basically of cellular respiration is to make more and more of these molecules, because this contains a bunch of potential energy, right? So when we have this molecule, we have a lot of energy being held with these bonds, and then once we break those bonds, we get that burst of energy. Okay, so we're going to go through for the next 2 videos this process known as cellular respiration. So this again can be thought of as a complicated process, but don't worry, we're going to break it down in detail for you. Okay, so we're going to, first of all, let's just study the chemical equation. Okay, so the chemical equation for cellular respiration, if we look at it, we start out with a glucose molecule. So glucose is C6H12O6. And we remember that from our carbohydrate talk when we were talking about macromolecules. So this is an example of a monosaccharide. Okay, so C6H12O6. Remember monosaccharides, they have that ratio of 1 carbon to 2 hydrogens to 1 oxygen. So this is kind of easier to remember. And then we have glucose is a reactant. We also have oxygen involved, okay? And then the products of this reaction are 6 carbon dioxide, 6 water, okay? So these are the byproducts, we call that. And then the whole purpose of this is to gain ATP. So we end up with, on average, about 36 ATP from this process. So in order to get 36 ATP, we need to have 36 ADP with 36 phosphates sticking around, okay? So you might want to write down this equation, just, you don't have to write down the whole thing if you don't want, but, just write down like the chemical formula part. But you can see it is a balanced equation. So if we look at how many carbons there are, we have C6H12O6. So that means that we have 6 carbons on the left side of the equation sign, okay? And we have 6 carbons on the right side, okay? So here's carbon dioxide. So we have 6 carbon dioxide, so there's 6. We have 12 hydrogens on the left side. We have 12 hydrogens on the right side. We have 6 oxygens on the left side, plus 12 here. We should have 6 oxygens on the right side plus 12 here. Okay, so everything should balance out in the long run. Okay, in this equation. So when we're going through this process, we're only going to focus in on this video, this part, glycolysis. The next video, we're going to focus in on the citric acid cycle, which used to be called the Krebs cycle. We no longer call it the Krebs cycle just because this is a person's name, and he necessarily wasn't the one that, there's other people that were involved in identifying it, so we don't just name it after one person. So we call it the citric acid cycle now. The electron transport chain, that's also involved. So this part we'll talk about in the next video. So this video will just concentrate on glycolysis. Now if you look at this figure here, glycolysis occurs outside of the mitochondrium. Okay, so the mitochondrium, if you recall from when we talked about the functions of the organelles, The mitochondrium is like the powerhouse of the cell. It's the little power plant. Okay, so glycolysis is happening outside of the power plant. So it's happening in the cytoplasm. Okay, but if you see here, it is producing a little bit of ATP. So we're going to talk about glycolysis in this video, but then we'll continue on in the next video, completing cellular respiration. Okay. So to get us started, we're going to watch an Amoeba Sisters video that goes through this whole process, okay, just to give us an overview of this whole process. And then we'll focus in on this part in this video and then this part in the next video, okay? But we kind of want to get like an overview first to just get us started. Okay, so we'll watch that.
Are you a morning person? One of us is, and one of us is definitely not. Mainly because when I wake up in the morning, it just takes a while for me to get my energy back. It takes a lot of time and coffee for that to happen for me. Cells really don't have that luxury. They are busy performing cell processes all the time. Active transport of many substances needed for their survival, for example. And the energy currency they need, specifically, is ATP. ATP stands for adenosine triphosphate. It's a type of nucleic acid, actually, and it is action-packed with 3 phosphates. We have a video all about ATP and how it works as an energy currency. So where am I going with this? Well, cells have to make this ATP. It doesn't really matter what kind of cell you are. Prokaryote or eukaryote, you have to make ATP. The process for making that ATP can be different, however, depending on that type of cell. One way is called aerobic cellular respiration. Lots of organisms can do aerobic cellular respiration, but this video is specifically going to go into aerobic cellular respiration in eukaryotic cells. That means this video is talking about the process within cells that have membrane-bound organelles, such as a nucleus and mitochondria. Eukaryotic cells include the cells you'd find in protists, fungi, animals, and plants. The mitochondria, which can be found in most eukaryotic cells, are going to be kind of a big deal in this aerobic cellular respiration because some of the process occurs in the mitochondria. So let's get started. Remember, the major goal for any organism performing this is to make ATP. Okay, here's the overall look at the equation for aerobic cellular respiration. Remember that reactants, inputs, are on the left side of the arrow, and products, outputs, are on the right side of the arrow. This equation, by the way, looks remarkably similar to photosynthesis. Look at how the reactants and products are on different sides. Well, that doesn't mean they're simply opposites. It does show that they have substances in common. In photosynthesis, organisms make glucose. Notice how glucose is a product. But in cellular respiration, organisms break the glucose down to make ATP. Fun fact, you know how when a bean seed first germinates in the ground, it isn't able to do photosynthesis yet? Yeah, the germinating bean is relying on glucose that it has stored, and it's doing cellular respiration to break it down to make ATP so it can grow. Of course, once it starts to mature and develop leaves, it can do photosynthesis. It will be able to do both photosynthesis and cellular respiration. Plants make glucose in photosynthesis, and they break it down in cellular respiration. They can do both. But if you aren't photosynthetic, such as a human or an amoeba, you have to find a food source to get your glucose. You need glucose to get this process started, and we're going to assume that we're starting with one glucose molecule. Step 1, glycolysis. This step takes place in the cytoplasm, and this step does not require oxygen. It's considered anaerobic. In glycolysis, glucose, the sugar from the equation, is converted into a more usable form, called pyruvate. Glycolysis usually takes a little ATP itself to start up. The net yield from this step is approximately 2 pyruvate and 2 ATP molecules, and 2 NADH. What is NADH? NADH is a coenzyme, and it has the ability to transfer electrons, which will be very useful in making even more ATP later on. We'll get to that in a minute. Now an intermediate step occurs. The 2 pyruvate are transported by active transport into the mitochondria, specifically the mitochondrial matrix. In the mitochondria, pyruvate is oxidized. The 2 pyruvate are converted to 2-acetyl-CoA, which will be used by the next step. Carbon dioxide is released and 2 NADH are produced. Step 2, the Krebs cycle, also called the citric acid cycle, still in the mitochondrial matrix. The citric acid cycle is considered an aerobic process. While the cycle doesn't directly consume oxygen, some of the events in the cycle need oxygen to continue. To learn more about this intricate cycle where the 2-acetyl-CoA will enter, check out the further reading suggestions in the video details. Carbon dioxide is released. We also produce 2 ATP, 6 NADH, and 2 FADH. FADH is also a coenzyme, like NADH, and it will also assist in transferring electrons to make even more ATP. Step 3, the electron transport chain and chemiosmosis. This is just a beautiful thing, really. In eukaryotic cells, this is still in the mitochondria, and to be more specific, it's involving the inner mitochondrial membrane. We do require oxygen for this aerobic step. This is a very complex process, and we are greatly simplifying it by saying that electrons are transferred from the NADH and FADH to protein complexes and electron carriers. The electrons are used to generate a proton gradient, as protons are pumped across to the intermembrane space. All these protons being pumped out into this intermembrane space generates an electrical and chemical gradient. The thing is, if you remember from our cell transport video, ions, like the H+, don't easily travel across membranes directly without something to travel through. The protons can travel through an amazing enzyme called ATP synthase. If I could be any enzyme, I'd be ATP synthase because it has the ability to make ATP by adding a phosphate to ADP. ADP is a precursor to ATP. ADP has 2 phosphates, but if it obtains a 3rd phosphate, it becomes ATP. So, in chemiosmosis, the protons travel down their electrochemical gradient through a portion of the ATP synthase, powering it to make ATP, the ultimate goal. Oxygen is the final acceptor of the electrons. When oxygen combines with 2 hydrogens, you get H2O, water. If you remember from our equation, water is a listed product. Now, the electron transport chain in chemiosmosis step makes a lot more ATP compared to the other 2 previous steps. How much? So I've noticed in my years of teaching and the assortment of textbooks I've collected over the years, there are some different numbers in cellular respiration charts. In fact, you can even see them change a bit in different editions of the same book. And it's made me want to emphasize that it's important to not just memorize a number, because it really is more of a range. I want to focus less on the number of ATP made per glucose molecule because it depends on a lot of variables. One variable is the gradient we were talking about. How many protons were pumped across the mitochondrial membrane? You can see more variables discussed in the factual references, and those references have estimates ranging from 26 to 34 molecules of ATP per glucose molecule in the electron transport chain in chemiosmosis step alone. Then, if you add the other 2 steps, Krebs, otherwise known as the citric acid cycle, and glycolysis, you could estimate a range of anywhere between 30 to 38 net ATP molecules total per molecule of glucose. Again, to emphasize a range. Now, this was just one way of creating ATP, but like we had said at the beginning, all cells have to make ATP, but the way they do it can differ. If there's no oxygen available, some cells have the ability to perform a process known as fermentation. It's not as efficient, but it can still make ATP when there isn't oxygen. We have a video about that. We can't emphasize enough how important the process of making ATP is for cells. For example, cyanide, which is found in some rat poisons, can block a step in the electron transport chain, which would block ATP production. A poison that prevents ATP from being produced can be deadly. With the important role that mitochondria have in ATP production, there is also a demand for increased research on mitochondrial diseases. We are confident that the understanding of how to treat these diseases will continue to improve as more people like you ask questions. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
So again, that gave us a good overview of the process of cellular respiration. But we're just focusing in on this first part, glycolysis. And as we go through these steps, you'll want to pay close attention to the inputs as well as the outputs of each of these steps, okay? So let's start out with this first step of cellular respiration where we're taking a look at taking glucose and breaking it down, okay? So glucose, well, what is glucose? Glucose is that monosaccharide, and so we get that from the food that we eat. So plants, where do they get their glucose from? They make it through the process known as photosynthesis. So plants, they're very self-sufficient. They don't need us, but we need them, right? So they can, they undergo glycolysis, they undergo cellular respiration, just like we do, but they don't actually need to eat to survive because they can make their own sugars through the process of photosynthesis. So we will bring in our own glucose through the foods that we eat. We can take it in as starch or, you know, as sugars that from the foods that we eat. And then our bodies will break it down to the monosaccharide form glucose, okay? So from there, we're going to be taking that glucose into our cells, and we're going to be breaking this glucose down. And as that glucose breaks apart, we're going to harness the energy by breaking those bonds, okay? Now, this process of glycolysis, both prokaryotes and eukaryotes perform glycolysis or some form of glycolysis. Therefore, we do think that glycolysis was something that evolved very, very early in life's history, since it, you know, occurs in bacteria and it occurs in archaeans. And so this is something that we do think, you know, evolved very early in life and is the way that early life forms harnessed energy. Okay. So it's a way that we break down glucose and we extract energy for our chemical reactions that occur. And if you notice, it occurs outside of the mitochondria. So our prokaryotes, remember, they don't have mitochondria, they don't have organelles. So this is occurring in the cytoplasm. So all cells have cytoplasm, so all cells can perform glycolysis. All right, now before we get into the process of glycolysis, we need to talk about electron carriers. And for some reason, your book does not have a separate section of this, so I'm going to take some time and talk about it so that we don't get really, really confused later on. So what are electron carriers? Now, we saw from the video how we have these little, well, they called them little coenzymes, NADH and FADH2, that like carry electrons to the electron transport chain later on. So we have these little electron shuttles that will actually bind to electrons and carry them to other parts of the cell. Okay, and so when we do have an electron added to a compound, we call that compound being reduced. Okay, so when a compound has added an electron, it's become reduced. Okay, when electrons are removed from a compound, it's considered oxidized. All right, so these are what we call, if you've heard the term redox reactions, if you've ever taken like a chemistry course, redox reactions is what I'm getting at here. And so we have these reduction oxidation type reactions. And it's just simply where we're adding or removing electrons. And these electrons, they get carried throughout the whole process of cellular respiration, which is why we need to talk about them. And we have two important molecules that are important electron carriers in this process of cellular respiration. We have something called NADH and something called FADH2. And you don't have to remember the full name. Okay, I don't even remember the full name. It's like nicotadamine dihydra blah blah blah. Okay, so it's a long name. You don't have to remember what it stands for. Just remember NADH. That's the reduced form. Okay, so that's when it has the electrons attached. Okay, when it loses its electrons, its form is NAD plus. Okay, so that's when it's lost its electrons, it's NAD plus. So when it gains its electrons, its reduced form is NADH. Okay. FADH2 is its reduced form. When it's lost its electrons, it's FADH. Okay. And these are what we call, again, redox reactions. One easy way to remember, like if it's reduced or oxidized, is the acronym OIL-RIG. Okay, so OIL-RIG, oil stands for oxidation is losing electrons, reduction is gaining electrons. Okay, so if it's reduced, that means that it has, reduction is gaining, it's gained electrons. If it's oxidized, oxidation, it has lost electrons. Okay, so that's an easy way to remember it. All right, so we'll talk about these electron carriers throughout the next few slides. So just, be aware for now what NADH and FADH2, that just means that they're carrying electrons. And we'll talk about when they're carrying electrons and so forth in the future slides. Okay? All right, so let's get into glycolysis. So with glycolysis, we can break it down into two phases. We have what's called the energy investment phase and the energy payout phase. So the energy investment phase is exactly what it sounds like. We actually have to add energy, or ATP, in order for us to break apart the glucose molecule. So here's glucose. Remember, glucose is 6 carbons, so it's a 6-carbon ring. You can think of it like that. So here's our 6 carbons, and we're going to have to add 2 ATP in order to break this molecule apart. Okay, so in order to break it apart, we have to add 2 ATP. And so by adding the 2 ATP, we break it apart, and we're going to break it apart into 2 3-carbon chains. All right, so we're breaking it apart into 2 3-carbon chains. So now we went from a 6-carbon glucose to 2 3-carbon chains. Okay. And in this process, we've now entered the energy payout phase. So in the energy payout phase, you can see what's happening. We go from this 3-carbon chain to an end product called pyruvate. So pyruvate is a 3-carbon chain pyruvate molecule. We've also noticed we've gone from NAD plus to NADH, so we've gained electrons. So this has been reduced because, OIL-RIG, reduction is gaining electrons. So we've ended up with 2 NADH, 1 for each pyruvate. And we've taken the phosphates and added that to 2 ATP. All right. So in the end, we end up with 2 pyruvate molecules, 2 energy carriers, NADH, and 4 ATPs. Okay. So if we end up with 4 ATPs, but we started out using 2, we have a net gain of 2 ATP molecules. Okay. So that's it for glycolysis. All right. So we have 2 phases, the energy investment phase. So we take ATP to break apart the glucose into our intermediates. And then the intermediates undergo the energy payout phase, where we end up with 2 pyruvate molecules, 2 NADH electron carriers, and 4 ATP molecules. Okay. So what I'd like you to do is, in your notes, create a chart. Okay, so in your chart, you'll want to have a column that says the inputs for one column and the outputs for another column. And then for a row, you're going to write glycolysis. Okay, so In your row for glycolysis, under inputs, you're going to write what the inputs were for glycolysis, and for the outputs, what the outputs were for glycolysis. Okay? And then that way, when we go through the next step, you'll add another row. Okay, so the next step will be it'll be an intermediate step, and then you'll write the citric acid cycle step and the oxidative phosphorylation step. So that way you'll create like a little table or a chart where you can summarize all of these. Okay, so again, I suggest that you have in your notes a little table chart. So you'll have the inputs column and the outputs column, and then a row for glycolysis. So the inputs of glycolysis, as we stated before, was glucose. Okay. And then the outputs, we ended up with 2,3-carbon pyruvate. Okay, so that's what happened to the glucose. We basically just split this C6H12O6 into 2,3-carbon pyruvate. Okay. We used 2 ATP, okay, to get this to break apart, and so we ended up with 2 ADP in the process, plus 2 phosphate groups. Okay. We needed 4 ADP plus 4 phosphate groups to form 4 ATP. Okay. And then we needed 2 NAD plus. And then using the electrons from the glucose, we added those electrons to the NAD plus to form the NAG carriers to NADH. Okay, so here is our outputs. And notice from the outputs, we ended up with a little bit of ATP as a net profit. Okay. So at the end of this whole process, if we only did glycolysis, we still gained 2 ATP from this process. So bacteria, they don't have mitochondria, so they can still undergo just glycolysis and get 2 ATP, okay, through that process. So this is a way that they can obtain energy. All right. Okay, so we're going to do a couple review questions. So energy is stored long-term in the bonds of blank and used short-term to perform work from a blank molecule. So energy is stored long-term in the bonds of ATP and used short-term to perform work from a glucose molecule. Or is it energy stored long-term in the bonds of an anabolic molecule and used to perform work from a catabolic molecule? Or energy is stored long-term in the bonds of glucose and used short-term to perform work from ATP? Or is it energy is stored long-term in the bonds of a catabolic molecule and used short-term to perform work from an anabolic molecule? So what is the correct terminology here? If you chose C, I would agree with you. So we store energy in the bonds of glucose, and then we break glucose down into pyruvate and extract ATP from that. And ATP is used for short term to perform work. Okay, so that's the answer. C, glucose, ATP. The energy currency used by cells is it ATP, ADP, AMP, or adenosine? So what's the energy currency? If you chose A, ATP, I would agree with you. So of course ATP is our energy currency. We use ATP and extract energy from it. So we break it down into ADP and phosphate, and a burst of energy is extracted. The glucose that enters the glycolysis pathway is split into 2 molecules of, is it A, pyruvate, B, ATP, C, phosphate, or D, NADH? So what is the glucose split into? If you chose A, pyruvate, I would agree with you. So glucose, the 6-carbon glucose, is split into 3-carbon pyruvate. Okay, so that's the answer. we do get ATP, but glucose doesn't split into ATP. Okay, that's just a product. All right, so that's the first part of the cellular respiration, glycolysis. So by now you should be able to explain how ATP is used by the cell as an energy source. You should also be able to describe the overall result in terms of molecules produced of the breakdown of glucose by glycolysis, okay? So in the next video, we're going to move into the mitochondrium, take that pyruvate, those 2 pyruvate molecules from glycolysis, as well as the 2 NADH, and continue on with the citric acid cycle and oxidative phosphorylation. Okay, so we'll continue with that video.